Bismillahirrahmanirrahim. Sexual reproduction in fungi. Our last topic, in our previous topic, we were discussing about asexual reproduction in fungi, and you know that there are 4 different methods of asexual reproduction in fungi. But today we will discuss about sexual reproduction in fungi, that how fungi reproduce sexually. So, sexual reproduction in fungi is simple than asexual reproduction. In sexual reproduction, as we know that there are 4 different group of fungi: zygomycota, basidiomycota, ascomycota, and imperfect fungi. But in imperfect fungi, the process of sexual reproduction is absent. It means that then only there remain 3 different group: zygomycota, ascomycota, and basidiomycota. In these 3 groups, the sexual reproduction has been observed. So in these 3 group, the sexual reproduction proceeds in different way. There is a slightly variation in the reproduction methods in them, but the thing that is common in all these 3 group is that there is a fusion of nuclei in all different group, followed by the process of meiosis in which spores are formed. So the process of sexual reproduction is somewhat different in all 3 group, but besides this difference, the common thing in all 3 groups is that there is a fusion of nuclei, number 1, and number 2, the formation of spores after the formation of spore by the process of meiosis. So that is the common thing in all these 3 things. We start from here that in sexual reproduction in fungi, that one is a long question. Fusion of nuclei occurs means 2 different nuclei come close together and they intermix to form the diploid zygote. And number 2, different in all 3 groups. But the common thing is that fusion of haploid nuclei followed by meiosis. Fusion of haploid nuclei and meiosis takes place in all groups. In all groups, this process takes place. Next one is process of sexual reproduction, that how this process is performed. The process is so simple that there are 2 different hyphae that is only genetically different, but compatibility means they are genetically different but they are compatible. They can intermix with each other. When they come close to each other, then first their cytoplasm fuse together and then their nuclei fuse together. First cytoplasm fuse together, this is known as plasmogamy. After this, the nuclei come close together, that is known as karyogamy. So between karyogamy and plasmogamy, or plasmogamy and karyogamy, there is 2 different groups. In some fungi, for example, zygomycota, the fusion of nuclei after the plasmogamy takes place immediately. But in 2 groups, ascomycota and basidiomycota, the fusion of nuclei after the plasmogamy does not take place. It takes too much time after the plasmogamy. So that is difference between them. The process is that hyphae of 2 genetically different but compatible, mean they are genetically different. There are 2 different nuclei. As we are saying that there are 2 different hyphae, it means that the hyphae are formed from the spores and spores are produced through the process of meiosis in sexual reproduction. So these hyphae are different. So these are genetically different but compatible, come close together. First their cytoplasm fuse, then nuclei. First their cytoplasm fuse, then their nuclei fuse. And if their cytoplasm fuse, it is known as it is the first step, and that is called as plasmogamy. Whereas in second step, that is karyogamy, the fusion of nuclei occurs. That one is a short question that differentiate between plasmogamy and karyogamy. Plasmogamy, the fusion of the cytoplasms, is known as plasmogamy, whereas the fusion of the nuclei is known as karyogamy. So in karyogamy, the fusion of nuclei, there are 2 different groups: basidiomycota and ascomycota. Karyogamy is late, mean in basidiomycota and ascomycota, the fusion of nuclei does not take place immediately after plasmogamy. In karyogamy, after the plasmogamy, there is a difference, or there is a time required for this process to complete. So in these two groups, basidiomycota and ascomycota, the karyogamy is late. That one is an MCQ. Acha, it means that if the karyogamy is not taking place immediately after plasmogamy, it means that now the hyphae are single, and in this hyphae two different nuclei are formed, two different nuclei are present. These nuclei not only present, coexist, they can divide within the same hyphae, and this hyphae bearing two genetically different nuclei will called as dikaryotic hyphae or heterokaryotic hyphae. But all this process we can summarize in a simple diagram, that this one is an one type of hyphae and this is an other type of hyphae. In this type of hyphae we represent it with an other ink, and this one is represented by a different ink, that you can understand that these two are genetically different hyphae. So these hyphae come close together, it means that their cytoplasm combine with each other, their cytoplasm fuse together, and when their cytoplasm fuse together, it means that it will form plasmogamy, that is plasmogamy. After plasmogamy, their nuclei also fuse. When their nuclei fuse together, then this process will known as karyogamy, or we called it as a karyogamy. This will known as karyogamy. Now the zygote is formed, zygote is diploid. Why? This hyphae was haploid, this hyphae was also carrying the haploid nuclei. When these nuclei will combine together, they form a diploid zygote. That zygote is diploid, and when this diploid zygote germinate and meiotically it will produce spores, and spores are haploid. As you know that during the process of meiosis, the number of chromosomes reduced to half. So number of chromosomes reduced to half through the process of meiosis. That's why from the diploid zygote haploid spores are formed. Now keep in mind that these spores are formed through the process of meiosis, and we know that in the process of meiosis crossing over takes place, and whenever crossing over takes place, it means that there is a variation in their genetic makeup. So these spores not resembling to this one and also to this one, but these are totally different from all their parents. Why? The reason is that they are formed through the process of meiosis. These spores whenever land on a suitable place or on a suitable position where moisture is available and food material is also abundant, then they can develop or grow to form a new hyphae or new mycelium. So that was all about the theory that how the hyphae or fungi reproduce through sexual reproduction. And in last, where karyogamy is not immediately, mean on those type of fungi, as we know that there are two different group, ascomycota and basidiomycota, where karyogamy does not occur immediately just before the plasmogamy. So the hyphae is the same, but it contains two genetically different nuclei. So that hyphae will known as dikaryotic hyphae or heterokaryotic hyphae. Where karyogamy is not immediately after plasmogamy, two genetically different nuclei coexist and divide within the same hyphae. This hyphae is called dikaryotic. This hyphae will called as dikaryotic hyphae or heterokaryotic hyphae. We will call it as heterokaryotic hyphae or dikaryotic hyphae. This one is also an short question, but here is an MCQ that a dikaryotic hyphae, he says that dikaryotic hyphae of two nuclei of different genetic makeup, similar genetic makeup, or any other option. So these two nuclei that resides within the same hyphae, if it is after the plasmogamy, then these nuclei are totally genetically different from each other. Next one is meiosis in zygote of basidiomycota will produce basidiospores. Basidiomycota, basidiospore, ascomycota, ascospore. But meiosis in zygote produce basidiospores, and meiosis in ascomycota also produce ascospore. Sometime produce means in some cases not at all. In some cases the spores of basidiomycota, basidiospore, and ascomycota, ascospore are produced basidiocarp and ascocarp respectively. Mean carp mean fruiting bodies are the reproductive structures, fruiting bodies that basidiocarp in some time basidia or basidiospores are produced within basidiocarp and ascospores are produced in ascocarp. That was a long question, and I try my best to make you understand that what is the correct way to write this question. And this is a very impressive and very explanatory, and it is easy for your examiner to check your question. So try your best to write in such a way that and inshallah you will get maximum marks in your exams. Allah Hafiz.